Good afternoon, students. Welcome to the class. Now, in today's class, we'll study about the development of fruit, apomixis, and polyembryony. So, first of all, we'll take up with the development of fruit. Now, what is a fruit? A true fruit is formed as a result of cell division, expansion, and differentiation in the ovary. The ovary is transformed into fruit as a result of stimuli received from pollination as well as from developing seeds. After receiving the necessary stimuli, the ovules present inside the ovary are transformed into seeds and the wall of ovary is transformed into pericarp. That is the fruit wall. The growth of the fruit is generally very fast, and yes, it is really difficult to define fruit because in common language a fruit may be defined as an edible product of the entire gynoecium and any floral part which is sweet, juicy, fleshy, colored, aromatic and encloses seeds. But however, the botanists they divide fruits into two main categories, that is the true fruits and the false fruits. Now, what are true fruits? True fruits, also known as eucarp, the fruit derived from ovary of a flower, not associated with any non-carpellary part, is called a true fruit. For example, mango, brinjal, banana, tomato, grapes, cucumber, pea, etc. These come under the category of true fruits. Right? And as far as the false fruits are concerned, that is the pseudocarp, the fruit derived from ovary along with other accessory floral parts. That is what is known as a false fruit. For example, in apple, fig, the main edible part of the fruit is the fleshy receptacle. Now here you can see in the diagram that of apple and strawberry. That thalamus becomes fleshy and that is the main edible part. As far as the horticulture is concerned, they call the term pomology to the study of fruits and its cultivation. So, if we take the term of horticulture for the study of fruits, that is pomology. Now, what is the role of pollination in fruit development? That is what we will study. We will study here the role of pollination in fruit development, role of seeds in fruit development, development of the parthenocarpic fruits and ripening of the fruits. So, first of all, we will take a role of pollination in fruit development. So, pollination is an important process essential for fertilization, seed development, growth of ovary and prevention of ovary abscission, that is dropping by the ovary. After pollination, the pollen grains release the wall-held proteins and other components. So, by this process, the recognition factors from pollen and stigma come close to each other. As soon as the pollen recognition is established, the growth in ovary shows abrupt changes. It is now well established that pollen grains release some specific hormones, particularly auxins, which together with the auxin of the carpellary origin stimulate the initial growth of the ovary. The effect of auxin in inducing the development of fruit has been observed in different plants like tomato, brinjal, tobacco, some cucumbers. So, next is the role of seed in fruit development. Developing seeds play significant role in the development of normal fruits. Why? because when seeds develop in all the sides, for example, in apple, if the seeds are present on all the sides, the fruit will developed will be normal. But if the seeds are present only in one side, then the fruit will develop on that side only. Is that clear? That is the relationship between the development of seeds and fruits. So it has been investigated that developing seeds, they produce auxins, gibberellins, cytokinins. These three are the plant hormones, the auxins, the gibberellins and the cytokinins, and they help in the normal development of fruits. Is that clear? Now seeds offer advantages to angiosperms. Firstly, since reproductive processes such as pollination and fertilization, they are independent of water, seed formation is more dependable. Seeds have better adaptive strategies for dispersal to new habitats and help the species. The hard seed coat provides protection to the young embryos. Being products of sexual reproduction, they generate a new genetic combination leading to variations. Now, seeds are the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seeds, which can be used as food throughout the year and also to raise crop in the next season. Just tell me, can you imagine agriculture in the absence of seeds? Or if seeds are there but they germinate right away, you can't store them, how difficult it would be. So how long the seeds they can remain alive? This time period varies greatly, and that is known as the viability of the seeds. So seeds of a large number of species, they live for several years or they may live for a few months. So some seeds they can remain alive even for hundreds of years, you know. There are several seeds which we have recorded, right? And the oldest is that of lupinus. This is excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. Now, a recent record of 2000 years old viable seed is of the date palm, that is Phoenix dactylifera, which is discovered during the archaeological excavations at King Herod's palace near the Dead Sea. So, this is the variation and viability of the seeds. Now, the next point which we have to discuss is the development of the parthenocarpic fruits. Some plants are able to produce fruits without fertilization. Now, such fruits they are known as parthenocarpic fruits. They are also known as seedless fruits because they contain empty or non-viable seeds. Now, some of the common examples of natural parthenocarpy are fruits formed as a result of ovary development without pollination. For example, citrus, banana, pineapple, etc. Or the fruits formed as a result of stimulation by pollination but without fertilization. For example, certain orchids. Or the fruits formed as a result of fertilization followed by abortion of the embryos. For example, grapes, peaches, cherries, etc. Some parthenocarpic fruits are artificially developed by the application of low concentration of auxins and gibberellins by some horticulturists. They develop seedless grapes, oranges, lemons, tomatoes, watermelons, etc. Why? Because these fruits, they have a great commercial value when they lack the seeds. Now, next is the ripening of fruits. After the period of growth due to cell division and enlargement, the mature fruit undergoes some qualitative changes referred to as fruit ripening. Some of the important changes during fruit ripening are softening of fruit flesh due to enzymatic degradation of cell wall, middle lamella, and solubilization of cell contents, hydrolytic conversion of storage materials into soluble sugars, for example, in grapes, apples, cherries, organic acid, for example, in citrus, oranges, etc., due to synthesis of new enzymes at the type of fruit ripening. Transformation of chloroplast to carotenoid rich chromoplast and accumulation of anthocyanins give characteristic color to the ripe fruits. Accumulation of flavoring components contribute to the flavor and aroma of ripe fruits. In certain fruits, ripening is accompanied by an increase in respiratory climacteric. A fruit ripening hormone, that is ethylene, also plays important role in the ripening of certain fruits. So this is about the fruits, how they are formed, how they ripe. We come down to the next topic, that is apomixis and polyembryony. Now apomixis, it is the development of reproductive propagules without meiosis and syngamy. For example, some species of Asteraceae and grasses. So apomixis is a form of asexual reproduction that mimics the sexual reproduction. Now if we talk about apomixis, there are mainly two types, that is the agamospermy and vegetative propagation. Now agamospermy can further be divided into three different types, that is adventive embryony, parthenogenesis, apospory and apogamy. So we take up these one by one. First we will talk about adventive embryony, that is the formation of embryo directly from the diploid sporophytic cells, that is nucellus or integument of the ovule, that is other than the zygote cell and is known as, for example, in the citrus. Then parthenogenesis, formation of embryo from the unfertilized egg. Apospory and apogamy, formation of embryos from any other cell of embryo sac that is other than the egg one without fertilization. So here fertilization is lacking. So during embryogenesis, an embryo develops from zygote inside the embryo sac and embryo sac becomes an endosperm. Now this apomictic embryo, if it develops, increases the number of embryos inside the seed. That is, if there are more than one embryos present inside the seed, then this process is known as polyembryony. For example, in many citrus and mango varieties, the nuclear cells surrounding the embryo sac start dividing, protrude into the embryo sac and develop into the embryos. These are the examples of polyembryony. Here you can see the apomixis. You can see the difference when there is reduction and recombination, that is the sexual division, and the sexual fertilization, formation of zygote, and here in the lower part of the diagram you can see there is no reduction and no recombination, and ultimately the plant which is formed, that is a maternal clone, that is formed parthenogenetically embryo. This is formed here without fertilization. The embryo is 2n. The above embryo is 2n after fertilization, but in the lower diagram the embryo is having 2n ploidy parthenogenetically. Is that clear? So you can see here in this table also, all these definitions they are given, and you can go through them. Go through this table please. And here you see the diagram of polyembryony, in which you see presence of more than one embryo. One is the zygotic embryo, and the here on the opposite side you see the antipodal embryo. So presence of more than one embryo is known as polyembryony. Now the last but not the least, I'd like to tell you that hybrid varieties of several of our food and vegetable crops are being extensively cultivated. Cultivation of hybrids has tremendously increased productivity. One of the problems of hybrids is that the hybrid seeds have to be produced every year. Do you know why? Because if the seeds collected from hybrids are sown, the plants in the progeny will segregate and they will not maintain the hybrid characters. So production of hybrid seeds is costly, and so this cost of the hybrid seeds becomes too expensive for the farmers. If these hybrids are made into apomix, here comes the use of apomixes. Then there is no segregation of characters in the hybrid progeny. Do you understand? Then the farmers can keep on using these hybrid seeds to raise the crop year after year, and he does not have to buy hybrid seeds every year. Because of the importance of apomixes in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of apomixes and to transfer these apomatic genes into hybrid varieties. That is the utility of the apomixes, you can say, in our agriculture. So with this we finish this today's lesson as well as this chapter. So we'll meet on Monday. So till then, please stay at home and stay safe.